A very good evening to one and all. Today is 16th August 2025, Saturday, and from 4 p.m. onwards, uh, this is the interdisciplinary series of POPMA Study Club August edition. And I am Dr. Bhuvan Nagpal, President of Practicing Oral Pathologists and Microbiologists Association, and I warmly welcome.
Sir, you are in
next
mute.
session. I don't know. So I warmly welcome everyone to the next session of POPMA Study Club Interdisciplinary Series. As an association devoted to advancing the academic, diagnostic, and research dimensions of oral pathology and microbiology, we believe that true progress lies in the embracing interdisciplinary perspectives. Today's session reflects just that. We are honored to host a speaker whose journey is not just inspiring but deeply transformative, a journey that began in the world of dentistry and now leads groundbreaking cardiovascular research in the United States. This session, titled Novel Role of NF2, Neurofibromin, in the Heart During Pressure Overload Stress, promises to be a compelling exploration of molecular science and its implication for medicine and clinical translation. On behalf of POPMA, I extend my sincere gratitude to Dr. Satvik Maridu sir for joining us and sharing his time, expertise, and story with us. I also thank our moderator, Dr. Nidhi Jadhav ma'am, for steering this session. I invite all participants, be it oral pathologists, medical professionals, or biomedical researchers, to not only learn from this session but to take inspiration from it. Let's today be a reminder that our beginnings do not limit our destinations. Let's dive into a session where science meets purpose. Thank you, and over to you, Dr. Nidhi ma'am, for the speaker introduction.
Thank you, Bhuvan sir. It's truly an honor to introduce today's guest speaker, Dr. Satvik Maridu, a remarkable individual whose academic evolution exemplifies curiosity, courage, and commitment to science. Dr. Maridu holds a BDS from Sivar Institute of Dental Sciences, Andhra Pradesh, and later pursued his passion for research by earning an MS and a PhD in biomedical sciences from Rutgers School of Graduate Studies, USA. He is currently a postdoctoral fellow at Rutgers, New Jersey Medical School, where his work focuses on understanding cardiac stress responses and the role of NF2 neurofibromin in cardiovascular function. But beyond degrees and titles, what makes Dr. Maridu's story truly extraordinary in his path is from being a first generation dentist in his family, unsure about his path, to becoming a first generation scientist. His career has spanned dentistry, stem cell biology, iPSCs, and now advanced cardiovascular research. He recently secured a prestigious postdoctoral fellowship from the American Heart Association, underscoring the impact and promise of his work. His vision is to one day return to India to establish his own lab and contribute to oral biology and translational research, particularly particularly focusing on conditions like oral submucous fibrosis that disproportionately affects our population. Today, he shares with us insights from his postdoctoral research on the regulatory role of NF2 in the heart under pressure overload. A talk that sits at the intersection of molecular biology, cardiology, and translational medicine. Please, all of you join us in welcoming Dr. Satvik. Welcome, Dr. Satvik Maridu, sir, and you can take over, sir, for your presentation.
Thank you very much, Dr. Bhuvan and Dr. Nidhi for the great introduction. And I would like to thank POPMA and Dr. Bhuvan and his team for this opportunity. Yeah, hope everyone will enjoy this. I would like to share my screen now. Hope everyone can see it. So you're able to see my full screen, right?
Yes, sir. It's visible.
Yeah, yeah. Okay. So as everyone mentioned, you can even have my contact if in case of any kind of like guidance towards research, I would be very happy to do it. I'm very much motivated towards research or willing to help anyone who is interested in uh, or have a similar passion. As Dr. Niti has already gave a kind introduction, I would just give few of the ups and downs I have faced personally. So from the beginning, right from my schooling, I am like little oriented towards science. For some reason, I'm more passionate, like how science helps in day-to-day activities. I had that driving, but however, as you know, the Indian society mainly focuses, especially in the South, I can strongly state, most of the careers are either towards engineering or towards medicine. And from a middle class family, I was like, had an option where I was suggested to medicine. And as many, I couldn't crack the medical entrance and ended up getting a admit in dentistry, which is a free seat and therefore continued over it. But however, I had opportunities during my undergrad where I planned to uh, present at various conferences. Like that was one thing which really helped me. And I still remember one of the conference that happened in Karnataka, that happened in Tirupati, but the president was from IDA president from Karnataka has come over there, and he said like less than 0.5% of research, even like research progresses in dentistry. This was back in 2008, I guess. So it was like very, very early. That really boosts me. Like, yeah, maybe I have to stick to research. Maybe I need to be one of the person in that very small percentile. And over the term, I was very much fascinated with stem cells, which was like pretty new concept at that point. I even presented in a conference of cancer stem cells that happened in Pune, where I was like challenged, yeah, I had to focus more on the coursework of dentistry because I am in the final years. But however, I took a chance and I went there, and I really had a great opportunity presenting over there. Then my only goal was, can we regenerate tooth? Back in 2011, I felt like why can't we have like a CAD CAM image where we could uh, prepare a tooth like a scaffold, and then have some stem cells and growth factors and grow as teeth. And with that interest, I felt like why don't I just feel like try whether I'll be a good fit for research or not, because I'm like not aware of any other things other than those Bendick tests or the physiology where you do the blood smears in the first few years of your dental school. Other than that, we hardly have used any of these laboratory techniques or tools. So I went to CCMB with a good reference. I was able to get a 3 month internship at CCMB, where for the first time it was like learning ABCD in research, like how to hold a pipette. The first thing I did was I guess many of you are oral pathologists, so you'll be aware of like milicu water, which is the distilled water. So I had to aliquot 1 ml of distilled water into Eppendorf's because I didn't had any prior experience. That was the first thing. Then slowly it moved to RNA isolation from stem cells. So the good thing was I was lucky and fortunate to be in a stem cell lab. And the research assistants over there, they used to give me like, okay, just start with two. Once I got a pretty good reading and minimal contamination, probably I feel like my surgical hand always helps me with most of the techniques. So it is more precise, and because you have that feather touch sensitivity of hand skill, I feel like that is a added benefit for me. Then I felt like little confident that okay, let me go over and try research. And then I came to US with a passion of doing stem cell research. But over the time, or maybe I could say that one of the oral big shot oral researcher at Rutgers, he said like we only do infection and we don't do any of stem cell work. Then I felt like little hesitant and I tried to start something else with the stem cell lab. Then I felt stem cell is just an application. I because having no background in research, I felt like maybe I have to start working on something basic science which always can be applied into multiple fields. So when that is when I did my masters. Usually masters with thesis has like one lab rotation and a thesis rotation, but I did three lab rotations and a thesis in a lab in order to gain the maximum expertise or experience, because we lack experience. So I felt like I have to spend every day in the lab so I can gain experience. And after that, I was able to get an admit immediately into the PhD program, where it was little tough because most of the students have research experience from their bachelor's, like biotechnology. You'll have some or the other coursework and lab. But being a dentist, we are not much aware of research. At least these days, people are the departments are training on writing these systematic reviews or some kind of writing experience. But I think back in early 2000s or 20 like 2010s, there is not much exposure on it, and it is I'm good that it is growing. I also feel it is important to learn such kind of basic techniques during the undergrad, because it's really really helpful. uh, because I would say like at least there will be one or two out of 100 of a batch that are interested in research, or who either with interest in dentistry or not much inclined towards dentistry who wants to move out. It would be a good opportunity. And to this I would like to add one more thing is, there are multiple opportunities in various central institute labs, as I mentioned, CCMB, like IASC or AIMS, or they usually have internships for students who are not graduated from the program. So I would suggest anyone who is in internship or in their final year to develop a project where they can always collaborate. And if anybody has any questions or want to ask something, I would suggest, I want to do this more like a discussion so they have an opportunity to interact. So please raise the hand, and I would think the moderator would be happy to chip in them. So then I felt like maybe I have to learn something more than oral biology. So for that reason, because oral biology is always my background from the dentistry, so I didn't want to study again oral biology. So that's the reason in masters as well as in PhD I never touched oral biology, and I focused mainly on mitochondria. I hope everyone is aware of mitochondria, which is called the powerhouse of the cell, where most of the metabolism happens, and it is the one which provides energy to the entire cell as well as the human system. And another interesting thing is, people always talk about oxygen, carbon dioxide, right? So most of the carbon dioxide conversion happens within the mitochondria, where the energy consumption happens, or where these redox reactions happens. So it is very fascinating that within a cell, inside an organelle, most of the things are being regulated. And I was very much curious for learning such kind of mechanisms at a molecular level. And then with my thesis, master's thesis lab, I drew a very much interest into the mitochondrial biology. And in PhD also, my lab rotations were related to stem cells and mitochondrial aspect of the cardiovascular disease. I did rotate with my chair, where autophagy was, he's a big autophagy person. But however, I felt like I need more interaction with my mentor, because I lack some background and experience. So I might need more focus from the mentor. So I chose a lab where I felt like the mentor would be uh, very much providing me guidance on a regular basis. And that lab was from Dr. Gopal Babu. He's a PhD from Madurai Kamraj, from Tamil Nadu, and he's been an amazing mentor. And I worked on Duchenne muscular dystrophy, which is a rare disease type, as you can see here. And I specifically worked on mitochondrial aspect, how the calcium imbalance in the Duchenne muscular dystrophy, and by regulating it, how it can help prevent the disease progression. And later on, due to my family reasons, I had to, my wife also works in the same department, and that's the reason I had to start my postdoctoral fellow in the same department but in a different lab, where I'm currently working on the project that was being outlined today, NF2, Neurofibromin. I think many of you people know about this protein in oral biology. Also, we come across neurofibromatosis, which is like type 1, type 2 disease, where I'll get around it, about it little later. And recently, as Dr. Nidhi mentioned, I did receive the prestigious American Heart Association Fellowship for 2 years, where I'll be receiving my entire salary from the AHA. So now I can do ask my boss, like, can you provide me some reagents, which where I can do some like cutting edge technology in it, and try to get some more data for my own project. And as you can see here, I did apply for fellowship twice during my PhD. So so American Heart Association is one of the fellowship for most of the international students, because we don't qualify for the NIH fellowships, the National Institute of Health, which is pretty easy to get, but only like you should be a citizen or a permanent resident in order to qualify for them. So this is the one of the opportunity, but it is highly competitive. But still, like out of 600 applications, probably 25% get funded, and it is like literally a big thing. Like there will be multiple stages. I wanted to give a few insights about the fellowship, because it would be nice if somebody is really interested in it. The reason I'm saying it is, most of the things in US you require a strong network. And I personally believe that after seeing multiple rejections, probably having a good network, people should know you, or your lab, that what you're working on and what your focus is. Because not every, just by looking at your application, it might not be possible, right? To judge a personality, or how his vision is. So at times it takes, you have to attend these scientific meetings and then talk to people, ask them for feedbacks. And another good opportunity I wanted to share is, we have a Society for South Asian Heart Research, where uh, I came across 2 years back. It is like 100 Indian cardiovascular scientists. That was like one of the first time where I had an exposure to see Indian cardiovascular scientists in that big number. And that was one of the good network where I have been connecting with and got a lot of feedback, and it helped me get my fellowship, I would say. And even the other contacts which my mentor has uh, initiated, like introduced me to his own mentors, where my current postdoc mentor did help a major role in structuring my proposal, as well as in initiating such conversations. And I personally feel, if you want to survive in US, networking is the key thing to either get a job, get an admit, or anything. It is not like merit, completely merit based, like in India, where, for example, you crack PG NEET and you decide where you're going. But here, in spite of grades, you have to have all other activities as a mandatory. That's how they judge your profile. So this is one thing I would like to give for any of those students who are interested in moving ahead. So now coming up to few insights I wanted to give. Like, what are the negatives of being a researcher, or what is the harsh reality? So like, as I wanted to compare to my own undergraduate batchmate, who like came over the same timeline. Like, I graduated in 2012. I tried to apply to a lot of PhD directly, thinking that I'll be qualified, because I don't know how many are aware. As a BDS graduate, you can apply to JRFs and SRFs, and you don't require a PhD. But however, those things are mainly only on paper, and I hardly see people taking it, because once I was being told by one of the chair at Anna University that I don't even consider you as a PhD candidate. So that's when I started applying for masters, where the red ones are usually the fee structure or the amount we pay. And most of my friends joined as public health graduates in either public health or health administration, and which is almost the same fee structure, but their route was pre-planned for a dental admit, where dental is very expensive. But once you graduate, you have to do like third and fourth year. And one thing I can strongly state is our experience as an undergraduate is way higher than any of the dentists over here, because we get a lot of OP exposure, which I think is very diverse, and where you will be dealing with uh, multiple like socio-economic regions, which will give you a wide variety of cases. I doubt everyone here will be getting the same kind of exposure, and I feel that we are more uh, so we have the ease in dealing with more challenging cases than anyone else. So I think our training is very rigorous, and the theoretical knowledge we have is completely different. One thing I can state is I still feel my biggest strength is my differential diagnosis. Even though I left clinics like almost 12 years, I still, when I have to like talk to anyone, I still have that systemic perception where I feel maybe this could be the possibility. And at least 80% of the times, I at least match with the diagnosis. That gives me a great confidence, and I feel that the place where I am trained is like really, really great. And as you can see, I started with a stipend of 30,000, which is like $2,000 a month, which is slightly hard for a family to live. But for as a single person, I can. So my friends started with this high DDS admission cost, and then they started in a private practice as a dentist, and their starting salary was 150,000. So it is almost like 5 times the difference. Like I qualified my thesis after 2 years, where you have to give a qualifying exam. But however, at that point, my friends started with a big package per year. And then it took me like with the COVID hit, it took like 6.5 years for me to graduate for a PhD. And my friends are by that time like an associate senior dentist, where their salaries, I gave an approximate, probably it could be even higher. And then my starting salary is 50. As you can see, it's always like 3 to 4 times. And now my salary is stuck at 68. But my friends, couple of them already own a clinic. And if you own a clinic, like it's a big thing. Like, yeah, it is hard to maintain a clinic, but still they earn a huge income, where like you can be having a luxurious life, I would say. Like, for example, if I'm bringing my parents, I have to think twice when will be the economy class tickets be cheap or something. But my friends, they need not, they can go for a business class ticket, where if they have to spare less than maybe three-fourth of their month's salary, they could get a business class ticket. So I'm trying to compare is research has big economic constraints. So you have to really be passion driven. Other than that, it is hard to survive. Yeah, in industry, you might be slightly earning better, but not as compared to a dentist. So now I want to give you why we have to do it. As I mentioned, it's a very low income, especially the academic positions. After 3 to 4 years of postdoc, you'll be eligible for moving as an assistant professor, where you make like 90 to 100K, but you have to constantly write grants, write papers in order to keep your profile getting tenured. It is not that easy because where teaching is like very minimal but research focused. There are multiple institutes, so it depends on which kind of institute, and also lots of sacrifice. One good example I can give you, for my wife's maternity leave, I took just 5 days off because I just started my postdoc lab and I had to be in the hospital. Those were the only 5 days I stayed away. So at times it is little challenging because these are my career junctions where I have to transition to the next stage. So I have to give my, so you will be having that guilt. Maybe I'm not giving my full extent for uh, the the research, or maybe I'm not giving full extent to the uh, personal family or personal life. So you will always not be able to balance the work and life. That is one of the biggest sacrifices. It is like how you have these emergency doctors. At least after they go home, they need not study about the current research. Maybe they have to, but it is not a very ongoing process. But in research, it is like, what is the next move you have to do, because everyone is like thinking 10 steps ahead. So you have to be like very 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 like proactive in keeping on learning. I would say like research is one entity where it constantly needs an update, like your phone which keeps on updating, or any software which keeps on updating simultaneously, is the research, which because I can consider even the update is part of the research. And another thing, there is no instant satisfaction like you get in clinics. Like you deal a case, I mean, a patient, and you get that instant satisfaction or a gratitude by easing their pain. That will give a lot of motivation, because I did clinics and now I'm doing research. I know the difference. So I always feel, what is your driving force? Like all these researchers, maybe at least 50% I spoke, they want to change their career because of their situation or work life balance or whatever you guys call. It is not that great. So many of them don't even want to pursue the same career if they go back 10 years back. So I feel like I am like very much like research driven, and that is one of the reason why I wanted to do. And as I mentioned, due to these low income in the academic, most of them go to industry. In industry, one of the major drawbacks is like you don't have the ease of doing what you want. Like for example, as Dr. Nidhi mentioned, my long term goal is to see, like everyone eats this betel nut in India. Like at least majority of the population has this betel nut abuse. What does it have impact on other systemic organs? Like very minimal research is being done, because all the studies that are there out are correlational studies where like they try to correlate, but there is the amount of basic science. Like for example, a mouse being given the betel nut for a while and then observing the systemic effects on various organs. Nobody has studied, or very few studies. Like oral submucous fibrosis, the number of studies on oral submucous fibrosis in the mouse are very minimal. So it gives us, it is important to understand the various mechanisms that happen, because most of the drugs are through these mechanisms, I would say. Like, why still choose research? If anybody knows, I would be happy to listen, because the motivating force is very very less, because you don't have great, like not uh, everyone can be satisfied in the research. It takes really long tenure. And can anyone say me, like, is research necessary or is it essential? uh, and I personally feel research is essential for existence of medicine, because if there is no proper research, for example COVID outbreak, if there is no proper research for the vaccination, probably it would have gone even more crazy. Therefore I feel research is very essential for the existence of medicine. Why I chose research? Simple thing. Like as a dentist, this is a number. Like I just gave a small thing. People even do like even camps and others. So I thought like maybe in a lifetime you might reach like 1 lakh cases. But as a researcher, not all discoveries lead to drugs. That's for sure, because that is the reason I was saying the satisfaction here is not that easy. And every discovery definitely leads to a new mechanism. A bunch of mechanisms together aids in the development of a drug. And one drug can like literally like if it is like a breakthrough drug, it can like uh, be saving a life-saving drug for lakhs of people or crores of people, because it can be used by everyone, right? So I feel, and for a drug it takes 2 to 3 decades if it is a very very concentrated. Nowadays I'm not talking about generic drugs where they try to change some of the molecular aspect and try to get a similar analog, but I'm talking somewhere where a new mechanism is being discovered and trying to target such like factors like transcription factors or downstream targets, and designing the drugs. So I personally have a question for, like, personally I believe it is mandatory to increase the training of physician scientists, not just the scientists. Can can anyone answer what could be the reason? That would be really nice. Like, how many people are really aware why I made this statement? I'm not like undermining anyone. Any of the basic scientists, their goal or plan will always be identifying a new mechanism. But how many people move to the next stage? As a clinician, I always feel the research should be translated. Not everyone gets the opportunity. So, for example, few people do MD PhD, which is like getting trained for MD and simultaneously getting trained for PhD. At that point, the PhD they do is not very rigorous as a traditional PhD. So their amount of research might be useful for translation, but not very much. They might not have complete idea about basic science. I feel like doing a proper traditional research will give you a perfect idea of basic science, and being a clinician, then you can develop something where it can be translated. And I feel that having more and more physician scientists would improve the clinical outcomes of the drugs, because you know what could be the side effects, because you observe them directly, and simultaneously you will work on the aspects where it needs to be changed or what could be the proper mechanisms. Like, for example, if you have any drug, most of the side effects are probably the liver toxicity or intestinal issues. So maybe you have to work on the absorption or something like that. How many are completely aware of the underlying mechanism or the mechanical stimuli that are being activated? Once if you know that kind of a basic mechanisms, probably you might think maybe targeting such molecules is should be taken with the precaution, and it should not interact with such kind of entities, therefore it might be helping. And this kind of knowledge, probably it's mainly I personally feel, I'm not like undermining anyone, but I feel that's my personal opinion, that physician scientists are like well versed with such a skill. And I think it is the time where there should be a increase in these people to bridge the gap between preclinical and the clinical research. And that is the driving force for me, because if everyone choose clinics, then who is going to do the research? Yes, it is painstaking and it is low income, but somebody has to give up on it and try to do the research. And another thing, I am like very much interested into clinics, but I chose research for a reason. That is mainly what I said at this point. So before moving, I wanted to give few techniques I learned during my tenure from master's, PhD, and postdoc. So one of the technique is I am now able to do echocardiography on mice, where we determine the cardiac function in order to understand how it goes up or down in the mice. So we use two modes. One is the M mode and other one is the pulse mode. The M mode is an ejection fraction where it determines the systolic function, and pulse wave, it determines the diastolic function. So this is one of the latest machine which we have. And I don't know if anyone is aware. It costs close to $1 million. So people have to write a very huge grant to get this machine. And they have to say like everyone in the department uses. It can't be used within just one lab. And this is the mouse heart. So showing the horizontal section of the heart, where these two are the papillary muscles and this is the LV, and here you will see an RV outlining. And this is when you draw a line, straight line across here, you will see this kind of bifurcation. And this is called the basic mode, and this is the M mode, where you measure in order to determine the ejection fraction. So cardiac function is determined by ejection fraction as well as the diameters, various diameters or LV parameters, left ventricular parameters. So this is outer wall, inner wall, and outer wall. So one could be the septal wall, because if you see this is the septal wall, and this is the right ventricle, and this is the left ventricle. So you need to, most of the people, because left ventricular function is major, so most of the diseases are left ventricular function oriented. People mainly use this data in order to determine. There is another mode called pulse wave, which is more resilient with the diastolic function. I can give you one example. So there are cases such as preserved ejection fraction, but still there could be a cardiac failure because of a failure in the diastolic function. And so these are two different types of Doppler. This one, where this is the left ventricle, and you determine the flow across the ventricle. And this is the valve, mitral valve, which you might have studied in your systemic biology or systemic aspect of the medicine. So when you're determining the flow, it is called pulse wave Doppler. But if you're detecting the displacement of the valve, it is called tissue Doppler. And these readouts mainly give you an idea about the cardiac function, like systolic as well as diastolic function. And because I felt this is like a completely new aspect to many of you people, I have so this is a software which I use to analyze, and this is one of the heart, where you can see. I hope everyone is able to see the pumping of the heart, right? Can someone confirm it, please?
Yes, sir, it is
Yes,
visible.
sir. It's
Yeah.
visible.
Okay. So this is the basic mode. And as you can see, the heart rate is like 500 to 600 beats per minute. And usually mouse heart, and this is under anesthesia. So I have a platform where I put the mice on the platform, anesthetized using isoflurane, and I try to regulate the heart rate. The heart rate should not be too low. So this is the basic. When I draw a perpendicular line like this, it leads to the, sorry. So this is the heart. So as you can see the earlier M mode, so this is the outer wall, inner wall, and outer wall. So this is how you draw lines. So there are tools where I have to hit then short axis view. This is called short axis view. As you can see, I drew a perpendicular line, and based on that, I calculate the dimensions. Got it? So this is one thing. And as you can see here, there is a good amount of peaks, right? Both upper peaks and lower peaks. Based on this, it determines the ejection fraction of the heart. And I can show you one more. So this is a mice which received surgery, but it is a wild type mice where there is no gene that is being altered. Now I would like to show you a mice that is knocked down for a heart. See, even though the heart rate is pretty high, see the amount of beating. It is not able to properly contract compared to the earlier, if you like see it. And I would like to see even the M mode. So see, there are hardly any visible peaks. You can see on one wall, but the other wall is really affected. And this mice, the ejection fraction will be significantly down, and by this we determine the cardiac function. So I wanted to give such kind of an overview, so it would be somewhat interesting to you guys. Apart from the pulse wave, I also learned confocal. I think most of you guys are well versed with confocal, because you guys are amazing oral pathologists with those HNE stainings, most of the case reports you provide. So this is an adult mouse cardiomyocyte, where I'm calculating the membrane potential of the mitochondria. So for example, you initially have a dye which is green in color, but with mitochondria, which has good membrane potential, these monomers become red aggregates and fluorescence. And based on the intensity, you compare between the wild type mice, the normal mice, and the gene edited mice. And this is a uh, temperature controlled Nikon confocal, where you can record multiple events, like over a time, how it increases or decreases. And then I want to show you, this is one of my favorite mitochondria, as you can see. So EM imaging of the cardiac section. And if you can carefully see, if you are aware of sarcoplasmic reticulum, this tubing like structure is called sarcoplasmic reticulum. This is 10,000x and this is 45,000x magnification, where you can see that mitochondria interact with sarcolipin sarcoplasmic reticulum, which have in tight junctions in order to mediate the calcium balance. So in heart, there is more of SR compared to ER, endoplasmic reticulum, because SR is the one which is essential for storing the calcium and contraction of the myocyte. And this is a beautiful image, and this is from my thesis paper from my PhD, where we try to understand the mitochondrial morphology between the Duchenne muscular dystrophy mice as well as the normal mice, and how on knocking down one of the gene, how it could enhance their health. And this is another instrument called seahorse, even though it terms as a seahorse is an actually an sea animal which is very minimal in size. But however, this instrument is mainly used to understand the metabolism of the cell. So you can isolate, like you can use cell culture in various wells, like as you can see in this plate, and then you can determine the basal respiration across the groups. And then these are various kinds of inhibitors that can regulate the mitochondrial respiration. So mitochondria is one of the key organelle for metabolism. So the first one, I know many might not be aware of the ATP synthesis that happens. So these inhibitors block various aspects, and based on that, you can find readouts. So this experiment is done in isolated mitochondria, but you can also use it in cells, cell culture, like using various cell lines. You can use primary cells or something where you culture cells in this kind of a dish, which comes with this, and then you take readouts and then quantify the oxygen consumption rate. So this is one of the, yeah, many of them have a lot of variation, and that's the reason you try to do at least like 3 to 4 essays in order to reduce the error. And now coming to calcium transients, because I work with those myocytes, like as I can see you here, these are individual myocytes where we isolate the myocytes from the heart, mouse heart. And then if you like put a small circular area like this and stimulate the myocyte with electrode, it gives specific transients. So this is nothing but release of calcium from the cell, which are being stimulated at 0.5 hertz, because these myocytes can be electrically stimulated. So this is, and once you give caffeine, most of you guys drink coffee in the morning, right? So it gives a huge surge, and that is the reason the cells are very active, or it gives you positive energy to work or focus more. So that is the reason what happens when a cell is exposed to caffeine. This is what happens. So lot of calcium is released into the cytoplasm, and then it can get into the mitochondria, which can boost the metabolism and make you more active. And not only the cellular, so here we try to determine the sarcoplasmic reticulum, how the calcium is released from the SR and how it is taken back into the SR. Based on that, we can get a lot of functional data, like how is the the calcium cycling happening within a cell. and this one is mitochondrial calcium. So I've been saying that I'm like very passionate towards mitochondria. So where here you see the regular stimulation, then we provide lithium thyroids, which is free of calcium and sodium. If you guys are aware, most of the myocytes have that action potential, so this one prevents such kind of contraction. And then you try to block the SR, because SR is the major, and then you activate the mitochondria, then you can see this small peak. And then you try to compare between the normal myocyte and the deceased myocyte, and then see the differences. These were various techniques I have been learning. There are a lot more. Like you guys are mainly familiar with ELISA. ELISA is more in a plate, but if you are doing the ELISA using an SDS page, it is called a Western blot. And other one is qPCR, which you might have been working on, because you try to do a lot of genes in the path lab. And now I wanted to give a mild overview of, like, because cardiovascular disease is, now I'm planning to show you what kind of research I'm doing during my postdoc. And with that I would end. I think it is a little longer today, sorry about that. As you can see, cardiovascular disease is one of the chief. And initially I am planning to show about myocardial infarction. Myocardial infarction is nothing but when people say that there are blocks in the heart, most of your vessels have been blocked, so that is leads to myocardial infarction or myocardial ischemia. And these are various statistics. I'm going to slightly brush up with this, and this is an overview how we do it in the heart. So for example, if you feel this is a mouse heart, so this is called LAD, or the coronary arteries, where you try to this is being blocked, and you try to use some kind of stents in order to perfuse this block. I don't know how many are aware. Most for example, if this is blocked and this caused the infarct or ischemia, if you kind of use stent, it actually increases the ischemia, because there is a sudden rush of all the molecules and oxygen radicals that are being generated, and it could actually cause increased expansion of the infarction. Compared to the initial, it actually goes up. That's why there are a lot of palliative care that is being provided in order to reduce this kind of oxidative stress. So that is the reason not every stent or surgery that is being done benefits the individual. In spite of providing vascularization, it still affects, because there is a lot of oxidative damage that happens. So that is the same thing which is similar to the cardiac transplant, where you initially don't have the supply and then start it. So therefore it is an important idea to study IR injury or ischemia reperfusion injury, because it has a great therapeutic aspect. And you can't do such studies in humans, and you need mouse models. That is the reason I was saying lot of the things people just, most of the doctors use drugs or surgeons do surgeries, but in order to understand mechanism, there should be a good amount of basic science research that needs to be done. So I'm going to give you a brief overview of NF2, the project which I'm working on. NF2 is a tumor suppressor gene. I think many of you people are known, and it is known to be an upstream regulator of YAP. YAP, or hippo pathway, mainly leads to cell proliferation or differentiation, or which activates the cell cycle system. And earlier study from our lab, which is the first study showing role of NF2 in ischemia reperfusion injury. I gave a brief introduction about ischemia reperfusion injury in order to stimulate, where in mouse you ligate the coronary artery, as you can see in this region, and then after 30 minutes you try to reperfuse, you release that suture and causing an ischemia reperfusion injury, and then you study various pathways. And in this pathway, we have seen that NF2 levels significantly go up, causing apoptosis and oxygen radicals, and thereby causing cardiac injury. And when you use a knockout mice to reduce the NF2 levels, YAP levels go up, and thereby mitigating the oxidative stress and thereby protecting the heart. This is a published research. Now we wanted to know there are different types of cardiac disease. So the disease we are focusing mainly is something related to hypertension, because that is one of the key cause for cardiac disease, where in normal heart, once you get hypertensive, there is an increased pressure in the vessels, leading to increased output, causing hypertrophy of the heart. So you can see the walls are thin, but however it becomes thick. But over the time, because heart can't meet the demand, it leads to heart failure stage, where the lumen becomes very big compared to the healthy heart, and the walls become really thin. This leads to end stage heart failure, and this is a different type, and you call this as dilated cardiac myopathy or cardiac myopathy in non ischemic heart failure patients. So therefore, this is another where there is an increase in the diameter. Initially, it leads to thickness followed by thinness. So this is how you do a pressure overload stress in the mouse, where in the mouse heart, so these are the aortic um, iota, ascending iota, where you block at the branches in order to create a pressure in the system and increase demand. Because there is reduced blood supply, there will be an increased demand for the heart to pump, leading to initially hypertrophy followed by cardiac failure. So this is a regular heart. After 4 weeks, you see there is a huge increase in the size, and if you cut this, the section would be something similar. So this is going to be the model which I am currently studying. So this is an existing project from the lab where I had to wrap it up, and we recently submitted this manuscript. And I would like to even state, we initially sent it to Nature Cardiovascular, straight away editorial rejection, then to JCI, Journal of Clinical Investigation. These are pretty good journals which has impact factor about 20. But however, they went to straight rejections. Now it is currently under revision at under review at Nature Communications. Probably you guys are have like maybe heard of these, but I'm saying like at times things are really hard. Even though it is there is a great amount of novelty, not everyone gets acceptance. So here is the research aim, where we wanted to study the role of NF2 in heart failure, mainly in non-ischemic heart failure leading to chronic stress such as TAC or transverse aortic constriction. So in order to first understand the disease during in a normal wild type mice, we wanted to see what are the levels of NF2 at 1 week of TAC, 2 week of TAC, 4 weeks, 6 week, and 12 weeks. As you can clearly see, excuse me, there is a significant increase in the first few weeks, followed by a decline, and by 12 weeks, which is the end stage heart failure, it is significantly down, suggesting that NF2 could be important maybe in the initial roles, but maybe due to some detrimental changes, it is reduced, but maybe it is detrimental. So we also looked at human heart failure samples. So donor means the person which has healthy heart, and recipient is the one which has the diseased heart. And as you can see, it is quantified here. The NF2 protein levels are significantly down because most of the recipients are end stage heart failure, and the mouse studies correlate with the human studies. Then we wanted to see like what could be the actual thing, what is the role of NF2, we wanted to investigate. In order to get that, we created a knockout model which is cardiac specific. So currently we are able to have this Cree-lock system where you can specifically knock out the protein in myocytes. So even heart, it's a complex. It has fibroblasts, endothelial, and immune cells. But here we are focusing mainly only on the cardiac myocytes. And this Western shows that it is a clean knockout. And as I showed you, we did cardiac function. So even in wild type, there is a significant decline. But however, in the knockout, you can see even more decline, suggesting that NF2 might be a really important protein, but however, on reducing its levels, it can kill the mice. or it can cause the disease progression in the mice. And we also do this kind of PSR staining, picric acid red staining, where we determine the collagen deposition and uh, quantify the amount of fibrosis. As you can see, compared to the wild type that went to the surgery, the NF2 knockout has a significant increase, as you can clearly see in these images. Similarly, we also performed RNA-seq. We always wanted to deal, as I mentioned, molecular mechanisms. What could be the pathways that are being activated in the wild type? And we wanted to compare with the knockout. So we determined a drastic difference between these pathways. And one of the top hit pathways is that metabolism is significantly impaired in these NF2 mice. And we thought ERR signaling, the estrogen related receptor, it is being significantly downregulated in the knockout mice. What are ERRs? ERRs are estrogen receptor related proteins. They are not the actual estrogen proteins. So these are orphan and they don't require the estrogen ligand, and they can activate the metabolism. There are well studied reports, like there are established reports stating that ERRs are known to activate metabolism, and they exist in 3 isoforms. Like for example, PDK4. Uh, Hope you guys are aware of the Krebs cycle, where acetyl CoA is a key entity for the start of the Krebs cycle. So the formation of acetyl CoA is a significant step in determining whether the cell should undergo glucose oxidation or fatty acid oxidation, and PDK4 is one of the determining enzyme. Why I'm saying more is we later used PDK4 as one of the enzyme to determine, and we isolated the lysates, and we saw that not only the RNA but even the protein levels of ERR alpha, gamma, and one of the key determinant for FAO are significantly down. And you can see this quantification. I'm rushing up little, sorry about that. We also used EM in order to determine the accumulation of lipid droplets. As you can see, these white spots are called lipid droplets, where in the sham you see minimal, which is a wild type mice, but after 3 days, the molecules are like little reduced, maybe because of the stress. But however, in the knockout, you can see these huge droplets suggesting an accumulation of lipids. We also did an assay where we used the the tissue lysates, and then determined using some kind of a colorimetric assay. And we saw that in both the knockout groups, it is significantly down. Based on this, we are trying to say that the fatty acid metabolism is significantly impaired. Then we were curious, does NF2 play a role in their transcription? But one thing we have to think is NF2 is more a scaffold or an adapter protein and lacks a DNA binding domain. Any protein that lacks a DNA binding domain cannot be a transcription factor. But we were curious, does it have a role, like indirectly? Then we first wanted to see whether NF2 can translocate from the cytoplasm to the nucleus, because most of the transcription happens in the nucleus where the chromatin gets like really active. So, and we saw there is a significant increase on TAC in the nuclear fraction of the cell. And this is an overexpression where in the nucleus we are able to see accumulation of NF2 protein. Then we wanted to do a GAL4 reporter assay. This assay is mainly done in order to show that uh, once you overexpress a protein, it will activate the transcriptional activity in the cell. And there is an increased transcriptional activity in on overexpressing NF2. Therefore, we wanted to knock down and overexpress NF2 and see luciferase activity for ERRE response elements. So why we do this is if you want to see that a specific transcription factor, is it activated? If a transcription factor is activated, its downstream genes are going to be increased or decreased. And we wanted to see on knocking down NF2, the ERR downstream genes are gone down, and on overexpression, it has gone up, suggesting that NF2 has some kind of a role in regulating these genes that are ERR dependent. And this is promoter pull down assay, where you prepare synthetic regions of the promoter, and then we try to incubate it with the cellular lysate, and then pull down using these streptavidin beads, and then run a Western. And as you can see, compared to the sham, means the mice which didn't go through the surgery, and TAC means the mice which went through the surgery, you can see a significant increase in the protein expression of NF2 for ERR alpha and gamma promoters, suggesting that NF2 does bind to the promoters, but it could be with a complex of proteins because it doesn't have the DNA binding domain. And using proteomic analysis, we identified Z-scan as the transcription factor, and later validated that Z-scan is the essential transcription factor that regulates these ERRs expression. And finally, we wanted to see, is ERR going to rescue the cardiac dysfunction in the NF2 knockout mice? So what we did, we injected AAV of ERR gamma into the mice, and then we did the surgery and then harvested the hearts. And as you can see, we'll focus on the TAC group, where we have seen wild type TAC has a significant decline, but these are the mice with just ERR in the wild type, and there is a good improvement in the heart function compared to the wild type. And this is the knockout mice, which is having significant decline, which I showed you even in the initial studies. And on overexpressing ERR gamma, the functional cardiac function has been rescued. And similarly, you can see the fibrosis, where you have the knockout and the overexpression, the fibrosis has been mitigated, suggesting that the gamma, which is a downstream target of NF2, has significantly benefited the cardiac function. Similarly, I have done the lipid analysis, which I am gonna skip at this point. So this is my concluding slide, where we feel that NF2 increases initially in order to meet the compensatory hypertrophy phase, because there is an increased demand, there is an increased metabolism required, therefore it is up. But due to the lack or failure to meet this demand, leads to lower levels of NF2, causing heart failure. Maybe we feel that lower levels of NF2, if it are being upregulated, maybe we could prevent the fatty acid oxidation impairment and thereby rescuing the cardiac function. So yeah, this is the end stage, and I would like to thank my lab members. So he is my current postdoc mentor, and this is my chair's lab, and these are the funding agencies that are providing me the support for this project. And I would like to thank you, everyone, for your patient listening.
Yes, Dr. Satvik, it
I'm
was
sorry, it took a long. I was like
no,
little fast. Sorry about it, but
uh,
yeah,
actually it was a blend of your journey and the topic and so many things. So it was indeed a wonderful and insightful lecture. Over to you, Nidhi ma'am, for further any queries and questions.
Thank you, Dr. Satvik, sir.
yeah.
Your
Go ahead.
research work tells your passion about, how much challenges you have gone through and how difficult the topic must have been. And you were also in the beginning very truthful about the details, the researcher, the challenges that one has to go through. What we as clinicians practice today is based on the findings that were researched, right, yesterday. But I think the problems that will come in future will be only solved when the research keeps continuing in the today's world. So no doubt that's very important. Both have to go hand in hand. Thank you, sir, for such a nice and insightful lecture.
I
Do we
wanted to
have any
give one
questions?
question. Can I once share the screen, or
Yes,
is
sir.
it
Yes, sir,
ended?
sure.
Yeah. Please go ahead,
Please.
sir. Please go
I
ahead.
have one last slide. One question. Do anybody know answer for this? This is one of the dream I want to see if anybody can do. I want to work for that. This
I
is
think
going to be the biggest dream I ever will have, but I would be like, at least before, someone who is working actively in some innovative field. And with this, I want to end the end the presentation. But I would like everyone to research: are there any dentists who have at least qualified for Nobel Prize? Qualified, like at least being nominated.
This it will be an inspiration to many of the dentists who are practicing this.
Yeah, that's what I feel. Like, as you guys can see, like Karanza textbooks or something for periodontology or anything, everything we see are correlational. Yeah, diabetes people have periodontitis, or like HIV patients, we can diagnose it just by looking at the gums. I was always fascinated with the clinics that happen, but how many are really having basic science studies where you support it? Everything is more like correlational. Like, one of the model I like, I'm proposing when I come back to India is high fat diet to the mice and simultaneously giving oral infection. So just by having just oral infection, there are reports stating that you might get the bacteria into the heart. There are like handful of studies, maybe like less than 10 studies, but people have done where they have injected bacteria or given bacteria in the oral cavity, and those traces are being found in the heart. But I wanted to use two hit model. There are people who have done MI also in the heart and simultaneously gave oral infection, and it was pretty bad. So means most of the comorbidities should be counted and then given oral infection. Then it would be a better aspect. So people like me should be more in number so we can study them better, like TB. There are multiple diseases that have cardiac function. Because I'm more in cardiac related, I will be thinking more on this. But I'm always want to you people to think also, like, because you guys have good amount of research, maybe you can state it to your research students, like your students, that uh, the significance of research. I think this is needs to be done more and more. I know people started stating about these systematic reviews or these public health surveys, but they are all correlational studies. It might not benefit. Like, the amount, for example, I'm facing a strong thing. I want to move back. I asked for a few dental schools. I don't have a BD. I just have a BDS. I don't have an MDS. And none of my masters or PhD are in oral biology. So I'll be just a senior lecturer, not even qualified for a bigger position. So there are a lot of setbacks which I was not aware of. Maybe if I had done MDS, people might have been taken me as an interdisciplinary scientist. But now I don't have option. And everywhere we see MBBS but not BDS. Maybe I feel like we have to strengthen that. I always believe the saying, oral health is the mirror of systemic health. And I think we need to do a lot of basic science research to support such kind of statements, I feel. So, yeah, with that, I would, and if anybody has any queries, they can always reach out to me, or you can use this podium to ask.
yes.
Yeah, I did see your statement, Dr. Bhuvan. Like, yeah, there are people who have been nominated, but we never reached to the
Yeah.
top-notch level, I guess.
So these are the only nominations only.
Yeah,
And
yeah.
the other study um, pertaining to tooth development for vitamin A and D. But he was a British physiologist, so
Yeah.
he was nominated for that one. Uh,
That's why I
yes,
see
I'll give.
being,
I have one thing, like the machines which you are telling, sir. They were specifically for the mice, or
yes,
they can be used for.
everything is for mice.
Okay, okay, because
Like, you can do it
humans.
for even human cells. For example, that you guys are aware of electrophysiology, right?
Yes,
So
yes,
those
yes.
myocyte calcium and all, they are patch clamp or electrophysiology instruments. So I have idea about how to do calcium. I'm not good with the patch clamp where you can see the currents within a cell or something. My wife has little bit of experience, but I don't have. Uh, so those you can use any cell types because we have muscles also, right? You can use muscle cells, like for example, the oral muscle or something like that. If you want to determine their activity, you can do that. There are a lot of other assays also. The cardiac echocardiography machine that you can use for mice and rat. So based on the platform size, because the probes are really small and mouse heart will be like tiny, really tiny. It's like the fist, right? The mouse fist will be like really small. So yeah, and confocal, like you guys are aware of it. The seahorse also, you can use any cell type. There are some protocols where you can use tissue sections. Seahorse is not very expensive. It should be probably around $100,000, which is like 80 to 90 lakhs or something. But the echocardiography machine is like really, really expensive because you can determine the blood flow. So you can also use it like contrast, like you can inject contrast into the mice and you can use it. Like you can even study vascular, you can study the tumor size. We mainly focus on heart, but there are a lot of other applications that can be done. So that is the reason when I'm looking back to India, at least I want a good mice facility, cell culture, and if they have echo, that is like more than happy. So that is one of my plan. When I'm moving, I always talk to my mentor. I said like, see, going back to India, I'll have good amount of human sample where I'll have access. So I'll be generating the human data, and probably you can use a similar mechanism in the mouse and then correlate, which will get us more funding and probably better science.
Sir, I think Dr. Kiran Jadhav wants to ask something.
Yes. And
Sure.
there is one more question
Yeah, one more
in the
In the
chat.
question.
chat also.
We'll take that also.
Hello.
Yeah. Hi, Dr. Kiran.
Yeah. Hello, Dr. Satvik. Actually, it was really nice to hear from you as BDS. After BDS, you have pursued the excellence in the research,
Mhm.
and really appreciate for sharing your personal insights. Actually,
Mhm.
now I just
Thank you.
want to know something about the mitochondrial DNA. Like how is the process of isolation of this mitochondrial DNA? How it's sensitive process?
I think I'm not much aware of just isolating the mitochondrial DNA, but what you can do is there are always copy numbers people use in order to determine the mitochondrial number. So you want to just only isolate DNA or you wanted to know specific genes?
Hey, for mitochondrial copy number, if we can just speak of about it, like how
So
do
mitochondrial copy number, it is simple. You isolate the overall DNA, which includes the genomic DNA as well as mitochondrial DNA. And then you use two references. One is for the nuclear DNA, and mitochondria has its own genome, right? So any of those uh, 13 to 18 uh, I'm not exactly aware of number of genes that mitochondria has. It's very less. So you can use any of those primers, and that's how you determine the copy number. Like R1IR, I should have some in my thesis paper because we did. One of the reviewer, like we quantified the number of mitochondria and everything. So they were curious what happened to the copy number. And it should be in my method section also. I can always forward it to you. You can reach out to me. I'm even sharing my email ID here. So if anybody has any, uh,
is this process is a very sensitive process, or like we
so
know
it
how?
is like how you isolate RNA. You
Yeah,
guys are aware of how to do a qPCR, right?
yeah. I've done qPCR for my PhD, so like I
Yeah,
have
so probably it should be the same. For genomic DNA, it is even more little simple because RNA can be degraded, but DNA should be very simple because it's more stable. But always I can forward you my paper with the method section so you can have a look at it.
thank you.
And I also see
I
one in
think,
the
sir,
chat.
one question is there by Dr. Abid. What
Very
kind of work would you recommend a BDS graduate to do if they want to
well.
judge whether clinical research is a good fit for them?
I think I stated that. I think CCMB was my first try, because see, at that point I was like literally worried, because the people I am challenging is BTech graduates, BSc graduates, MSc graduates, and MTech graduates. Those who are biotech or any of the sciences like biochemistry, all of them. I'm competing with all of them. At least in dental fraternity, I have crossed all of them, and I'm challenging only dentists. But this is even a bigger pool, like M Farm, B Farm, all of them come into the action. But I would suggest, if the person is still not graduated, to go and apply these programs, these institutes like any ICMR or CSIR institutes. They have these internship programs. So if you are part of a institute, you always have an easier entrance. Once you graduate, the chances are very less, and you might need a strong reference, internal reference, and probably go to a lab which fits your research. And most of them try to really support you guys, because they are not providing you a lot of. Only thing is, when you write, you have to say, I'm giving a good amount of time to them, because they have to invest their time in order to train you. So you have to really show, I just want to do for a few weeks, I just want to do for a month. What are you going to learn in a month? You need at least few months, like 6 months, I would say. Then you can do a small project where you can showcase it in any of your conferences or something. That's how they know it. And then you can also get a letter saying, because they know what you have performed in 6 months, how progressing are you in 6 months. So clinical research, there are two different things. What I was talking is about basic science. When you mean by clinical research, any of the public health research is clinical research, because it is linked to human subjects directly. But how many really do human basic science research? It's going to be very less. You have to identify. Uh, I know one person, Dr. Ullas Kultur. He's CDFD director. He earlier worked in TIFR, Bombay, then in Hyderabad, and now he said, like how I said about metabolism, right? There is a chamber called metabolic chamber, which determines the oxygen and carbon dioxide intake in a day and calculates the expenditure, energy expenditure. So we do that metabolic chamber thing for mouse. He said they do with CMC velour for humans. Nowadays, metabolism has been a big game changer for understanding the disease pathology. See, the things are being advancing, but being a clinician, we are not aware of such things. And specifically focusing on oral, we might not be aware of things. Maybe you guys have to first delineate the interest, because you can't just do anything and it might not be of your interest. Then you might not get motivated. Maybe cut down to the interest, and then think about where you can learn little bit of the techniques you wanted, and then apply for it. Not every lab does clinical research. For example, a lab technician in any of your pathology centers, that is part of a clinical work actually, because you're directly processing the samples. But in order to something higher, then I would suggest go for a basic science lab, something in, like as I mentioned, any of those DST, DBT funded, because they have certain funding. Wherever they say MBBS graduates, it is a hyphen BDS graduates. They don't use the terminology, but they indirectly imply it. Uh, yeah.
Yeah.
Thank you.
Good evening, doctor.
Hi, Abhishek.
I'm Dr. Abhishek Vedrina. And I don't know, recently I met Dr. Santosh, your periodontist, who happens
Okay.
to be a teacher.
He's in the southeast, like Arab.
Kuwait. Kuwait.
Yeah.
He had
He
come
was
down to
my
Bangalore.
back in my undergrad. He was part of our
Yeah,
department. He did train us actually. So
yeah,
I did meet him a couple of times, but very happy that he's.
yeah. So I happened to meet him on Sunday, part of one conference of the digital dentistry. And I was just speaking about research and oral pathology, and he spoke about you. Okay. And I was attending your lecture anyways, but I didn't know you were the same person. So happy to have seen you and seen your work actually. It's excellent. And he was actually looking out that you were looking at something in India. So there is this IASC Medical College coming up. Okay. Tata ISC Medical College, okay, where they're providing this postgraduate programs only, like it's MD, PhD programs, where they provide MBBS graduate with MD and PhD or MD, MTech, MS, MTech, or MS, PhD programs, where as you said, clinical scientist. Okay. So there in that I happen to speak about uh, why not have a MDS PhD program? They said like we don't want to offer MDS PhD. So I said, sir, you should. I wrote a mail that saying that you should not miss the train like the whole of other people do, where they keep the oral cavity aside and do research for everything else. So I have sent some contacts to Dr. Santosh. I think so. He thought he will share it with you. I'll share it to you on your mail also. Okay. I think so.
So
You should
just
write
to
down.
add on that aspect, I did go to their New York meet. So IAC actually traveled across US in order to gain a lot of physicians to India. So they are really welcoming a lot of MDs, but not any dentists
It's
or
so sad.
basic
So
science
sad.
researchers. I did speak to them in person. At this point, they are focusing only on MD. The reason is because they have a medical school, they want to train the doctors because we won't be qualified for them to teach any of their subjects. So they said like they want only MD biochemistry or something. But actually speaking, we also study the same way how they study it. The curriculum
Yeah.
is same, but yeah.
Yeah. So what I say, I have written a mail saying that you should have a oral health research department where you don't give them
Probably they said once they have the dental school plan or something, they might think of it, but
Mhm.
they straight away said it as a no to me. But
Uh-huh.
yeah, it is always because IASC is one institute which is a top-notch school in India.
Yes, yes, yes.
And
And
if you come to a basic science institute, NCBS TIFR is the top notch. But if you are coming to CSIR, CCMB is the top notch.
mhm, mhm.
The facilities I am exposed in 2013, I can compare that to my school education in Rutgers.
That's good.
So it
Okay.
is that well established. And the reason
Mhm.
why CCMB is that great because CSIR was once at Hyderabad and that was the CCMB. It was
Mhm.
later on moved to Delhi. That is the reason it was built very well.
Okay, doctor.
And I think the earlier director Lalji, he was an amazing person, and after that things were different. But he has made it a really big school. So
Okay.
yeah, I am like that always hinders me. Now also I can get a job in private school, but the issue is brand, right? For example,
Mm.
nowadays I think the salary structures are the same between government and private. It is not any different. But at the end of the day, if I'm graduating my student from IISc or CCMB versus SRM, WIT, or some other private institute, it's a big difference
Yeah,
because
yeah,
IITs
definitely.
are still valued.
Yes, yes.
So and other reason why I want to focus on CSIR or something like that is they have stipulated amount of funding which many
Mhm.
people are not aware. You need not even write amazing grants.
Mm.
All the funds are allocated, and if you write a decent proposal, you'll get it.
Yeah, okay.
So these are the things which we are not aware of. And as I mentioned, I'm waiting for a day where I want to have a flyer stating at the bottom BDS also in the flyer. Those who are BDS graduates and willing to study PhD, yes, I'm gonna open. So that is what I want to build a platform. It is easy for me. I stayed in US for more than 10 years. I came in 2014. But now because of this, I wanted to like come back to India because it's going to be harsh. The amount of research, I might not be able to do it. Many people suggest don't go, but I want to go because I have heard a lot, because still we are undermined being a dentist. If we don't step up, then nobody else is going to step up. And even at this point, I can move to pharma, but I still don't want to move whatever financial constraints I have. The only reason is, if I change my stream now, whoever still feels me as a success, which I'm still not a successful story yet, unless I graduate my first PhD student, I don't feel or qualify myself successful. Once I have one student out of my lab as a PhD, then I feel I'm like, and if it was a BDS graduate, I'm like, yes, that's my the biggest achievement for me. I'm
No,
I
for all of
I'm
us,
eagerly waiting for that.
yeah, for all of us, you're already a success doctor. Like you're
That's
following
what I'm
your
saying. But that is not
following your
successful to me, because I wanna have something for the dental fraternity,
That's so nice, doctor. That's so nice.
because we lack that
Yeah, yeah.
personally. Yeah, but
I think so. Dr. Bhuvan would really connect you to the right people. There is something called APJ Abdul Kalam incubation for dentists or dental
Yes,
this
yes.
one, and he is
No,
connected with.
sir, similar kind of things, no, I've written letters representing to ICMR. Now they have considered BDS, but still that mindset, which is like the discrimination part, it will
forget about
it will
BDS,
take time, sir.
sir. Even
There are
MBBS, they are not willing. No basic scientist is willing to train any physician. In US, the mindset they have is even terrible. They feel that research is not great. And many people, doctors, they think little bit undermining on the BDS. No, sorry, research itself. They feel they come at leisure. There are few students who do really great, but many students, they do it for a time, like because they have to improve their profile if they have to move ahead in their career. So there are multiple reasons, but still, yeah, I want to do what I can do, because it has to go step by step. I think it can't be done in a thing, but definitely I
yes,
hope
yes.
things are
That
changing
is.
slowly.
Anyway, sir, it was nice interacting with you.
Yeah.
So, um, thank you
Yeah.
so much, sir,
And
for.
yeah, I'll be happy if you want anything for the students alone, because I'm always happy to talk to them. But definitely, and thank you very much for this, Dr. Bhuvan.
Yeah.
Uh,
So, thank you so much, sir. And a small token of appreciation from the POPMA governing body, because we conduct this initiative of POPMA Study Club. And thank you so much, sir. It was such a wonderful session and it was very
hope at least it
inspiring.
was useful for
And
you.
definitely, students would have appreciated. I know this time it was actually a long weekend, and today festival is also there. But
Yeah,
definitely,
Krishnastami, right?
people will
Today.
see the
So
recording. If anything is there, we'll definitely
yeah,
connect with you.
as I mentioned, I gave my email ID. You can even include into your YouTube
Yeah, yeah. I
thing
will. I will share
as
it,
a
sir. I will.
timeline. So
Yeah, yeah.
I'm more than happy. And once I move back, yes, definitely I'll be connecting to many
Yeah,
of
yeah.
you people, because I want to do oral studies also.
Definitely, sir, and will take your help in selecting to the institutes. We will write on the platform of POPMA and represent that BDS should be included, because even our primary qualification is BDS and then for oral pathology, and the major chunk of research is done by
Another thing,
all the branches, but the kind of what should I say, the oral biology and oral pathology, all those molecular things are there. The lab part which is being associated is
because
more with
one
us.
thing is, sir, many of them I have noticed are doing PhDs, but many are doing only biomaterials. So that is the little constraint I feel like, why not trying little ahead? Because I think US is good in that aspect, where I know one dentist, she has done masters in computer science.
Mhm.
Dr. Sanjana, I guess.
No, no.
Like,
There is a person who has studied MTech also, or is doing research uh, at NIT or IIT. That is also there. I'll share that
yeah, but the
story.
numbers are slowly growing. I'm happy
Yes.
about it.
Yes.
But
yes.
people have to think in that aspect where as a dentist, we are not constrained to just oral biology. We have to get to other systems so we can correlate. Multidisciplinary, this is the era of multidisciplinary, I feel, and I think it needs to be reinforced.
Thank you, sir. Thank you so much for your time
Yeah.
and
Have a great
sharing your views.
and happy
Okay.
Janmashtami, everyone, I guess.
Yes,
I
yes,
think
yes.
everyone left, but I think I wanted to
Yeah,
wish
yeah.
you. Thank
So
you all.
we'll take a leave and uh, we'll see on 19th of August with another session of
Sure.
POPMA Study Club. 19th of September. So Dr. Mandana madam, she will be there and she'll be delivering it. So stay tuned.
Thank you once again for the
Yeah.
opportunity, guys.
Thank you, sir. Okay. Thank you so much, everyone. Bye.